Thank you very much, Jason. So, like you said, my name is Michael Opere, as you can see there, and I'm from Kenya. And mostly, I think when you hear Kenya, then you link it to athletics, because that's what our main export. The last time that there was a world championship here in Budapest, we harvested several golds and silvers, and we're among the top somewhere there. But for me, I should say that I've been running a different kind of race. I have not been chasing medals and silvers and golds and things like that, but I've been chasing viruses. And today I want to present to you part of the race that I've been doing, race of chasing viruses. It's about viral ecology. And because viruses are very dynamic, I will begin by explaining something about the ecological aspects of viruses in Lake Victoria, which is one of the largest lakes in the world. And then I will transition to looking at the viruses in an engineered system, following the treatment systems of the water treatment works that we have around, with respect to a treatment system in the United Kingdom, where I did a research following viruses on how they partition within the treatment works. So to begin with the ecological aspects, it's important maybe to understand the general biosphere in the world with respect to the global biosphere. And viruses are the most abundant biological entities on Earth. As you can see those figures there, it's to the 10 raised to power 31 particles. That is quite big, isn't it? It's like saying that the viruses are more than the stars in the skies, and they are more than the bacterial cells in the world combined with archaea and maybe even eukaryotes in total. So that's quite big. And then when it comes to the waters, or the environmental waters for that matter, then the ratio is about 10 billion particles per milliliter of solution. Again, that is very big. And this is not just numbers here. It's saying something. It's telling us that actually viruses are one of the ecological dominant entities in the world. So it speaks about ecological dominance. And then a large portion of these viruses are enteric viruses. Enteric viruses are viruses of the gut. Those viruses that infect the gastrointestinal system, and they cause a myriad of diseases related to gastroenteritis, as well as certain non-enteric viruses and non-enteric diseases like hepatitis diseases. So we have certain enteric viruses that also cause hepatitis. For example, hepatitis A virus or hepatitis E virus, they are part of this group of enteric viruses. Now, as to why these viruses matter when it comes to ecological aspects or water for that case, it is because the enteric viruses are generally non-enveloped viruses. When we categorize viruses, we categorize them as either enveloped, one of the categories, or non-enveloped viruses. For the enveloped viruses, of course, they have the lipid layer or lipid envelope surrounding the capsid, which is the protein coat that encloses the viral genome. So the lipid envelope is actually an Achilles heel, for that for lack of a better word, to the viruses, because it exposes them to vagaries of the environment. That means they're exposed to degradation, they are exposed to chemicals in the environment and the likes. But for non-enveloped viruses, they don't have the lipid coat. So as a result of that, they're mostly persistent in aquatic environments, and that is why they are viruses of interest when it comes to water. Then, with that, it's suffice to say that these viruses are very infectious in low doses, to the level that if they have less than 10 particles, and maybe ingest less than 10 particles, then that is enough to cause ripple effects in your immune system, and of course, generally will cause sickness. So they are very infectious at very, very low doses. Something that's very critical when it comes to consumption of contaminated water and food, and exposure to non-sanitized conditions. Then it's important to talk about these viruses with respect to freshwater systems. And that's where the Lake Victoria concept comes in. Why freshwater systems? It's because the freshwater systems are linked, it's like a holistic kind of ecosystem where there's a lot of interactions between animals, humans, and the pollution dynamics. Here we talk about, we introduce the concept of One Health, where a large lake like Lake Victoria, for example, is a meeting point or a convergence zone for pollution, animals, human beings, and so there are all manner of pollution pathways that may lead to these viruses ending up in such kind of ecosystems. So it is important to understand the lakes and rivers and other freshwater systems as transport corridors and temporary reservoirs for these kind of viruses. Then now on to bring it into perspective so that we all understand what we are talking about. This is a table of the most common enteric viruses. And in particular, I want to draw your attention to the first row. That is where we have adenoviruses as one of the enteric viruses. I put it up there intentionally because this virus is a special one among the enteric viruses. Why is it a special one? If you look at the column of the genome column, that is the column where we have the classification in terms of the genomic aspects of the virus. And the genomic aspect is very important when it comes to stability in the environment. Why? As you can see, adenovirus is a double-stranded DS, that's double-stranded DNA, and that makes it very stable. It's not only DNA, all the other ones are RNA viruses. And of course, the virus can either have, they only have one. It's either you have a DNA or you have an RNA. So we either have RNA virus or DNA virus. So for this particular virus, the DNA virus, that one makes it a classical resistant virus in comparison to the rest. And besides that, it also have a double strand, which means the genome is double-stranded DNA. And so in compared to the others that you can see SS single-stranded, then it means it's very, very stable in the environment. And that makes it, if you look at the column for environmental persistence, then you can see we are saying that it is it's resistant to chlorine. And when it comes to water treatment, then what comes into your mind is chlorine as a disinfection stage. And so this virus is resistant to chlorine, besides being environmentally stable. And then on the last column, we talk about the geographical patterns and endemicity, that is. So this virus is quite ubiquitous globally, which means you can find it everywhere. And it is because of the stability, so it can transcend several climatic conditions and it can survive in diverse sets of climatic conditions. Then it won't be good if I don't talk about enteroviruses. I'll skip noroviruses. I'll talk about enteroviruses basically because this is it has got several viruses combined together. It's a term for several viruses. As you can see there, we have Coxsackie virus, we have ecovirus and poliovirus. Of course, we know that poliovirus, we have the neurological conditions causing paralysis. But basically what stands out in this virus that it is enteric virus. Again, and there, of course, the others as well. And the reason as to why I'm saying it's also important to talk about this is because these viruses are the viruses, it's like the king, we talk about the king of the flus and colds and, runny noses and all that. So these are the kings of that particular kind of infections, quite acute but very significant. Suppose you understand that. And of course, so talk about non-enteric polio is the one that causes the neurological conditions, but the enteric one generally causes gastrointestinal illnesses. Very, very important. Then rotavirus also very important one, in the sense that rotavirus, as you can see, there's a double-stranded RNA virus. Very important because, as you can see, the others are single-stranded RNA, but rotavirus is double-stranded RNA. And why am I pointing it out? I'm pointing it out because of this particular, let me use this pointer. It's a nice one here. So I'm pointing it out because of this particular area. It will become more realistic when we're discussing the partitioning of viruses in the treatment works. So it can percolate and remain suspended in solids and even the sludge. Probably because of maybe there's some elements of attachment that is interesting as far as its genome is concerned. And it's a killer to children. This is a killer to children, especially in low resource content settings, for example, in sub-Saharan Africa and other areas in the world with, low socioeconomic areas. Then, as for the hepatitis A virus, I also mentioned that. I can't leave that. Why? Because of this. As you can see here, this particular one is also zoonotic. And when we talk about zoonotic viruses, because what should come into your mind is the coronavirus, the COVID-19, and all the conspiracy theories about this. Because we are not just sure which other virus would jump from animals close to us and cause another pandemic. You know, the coronavirus has struck a reminder that the world almost became to its knees because of the fact that we interact with animals, something very interesting. And because hepatitis E virus is zoonotic, of course, we interact with pigs, we rear pigs, and then, of course, the deer, and so this is one of the enteric viruses that is zoonotic. But there's a potential for all these viruses transforming themselves and mutating and becoming zoonotic in a way, one way or the other. And so one of the convergence discussions at the moment is about the zoonoses and viruses. Enough for that table, so I would proceed to maybe the current issues and emerging trends in environmental viromics. And so we have a lot of interesting things here that probably inform this kind of research. The first one is wastewater-based epidemiology. And wastewater-based epidemiology is one of the current trends in water, in virus surveillance in the environment. Because with COVID, there was a lot of virus surveillance in the wastewater aspect, because it's found that these viruses, the coronavirus, can also exist in the wastewater. So why is this important? This is important because we need to track the viruses real time. And of course, at the moment, there are activities going on to discover sensory technologies where we can be able to track viruses real time in the environment. This will augment the clinical aspect and will help in maybe foretelling the viruses that are circulating in the community without necessarily waiting for the clinical signs and maybe going to the hospital and all that. So we can be able to tell something about the viruses circulating in the community. Real-time tracking, very, very important. So it tells us about the hot seasons, hot spots, when we use wastewater-based surveillance, so that we can be prepared. Early warning signs for proper recourse. Then again, the wastewater-based epidemiology is also telling us about viral persistence. And for us to be keen on the risks, we need to understand the persistence of viruses. All these viruses talking about how do they persist in various environmental conditions. Very, very important. And we see that with wastewater-based epidemiology. And a classical case of example of what wastewater-based epidemiology can do, you can see it here with this map, where we have, this is human, this is hepatitis A virus, I should say. So this was a research about hepatitis A virus, and it is mapped all over the world with respect to wastewater-based epidemiology, research carried out in 2012. So this is just an example on what we do with wastewater-based epidemiology. Very, very important. Then, again, climate change is another area of concern today. And climate change is an area of concern because a lot of things are happening. Even though some people don't believe in this, like Donald Trump, but it is something that is, to me, I think it's realistic. And to me, I don't look at climate change as maybe change in patterns of weather. I look at it in terms of virus. What does it do to the viruses? Because that's where the rubber meets the road for someone like me. So climate change could influence stability of these viruses. For example, temperature changes, changing rainfall patterns, they could influence the stability of these viruses, and then, of course, other dynamics will come at play, for example, mutation, and we can end up with more potent viruses. Then also extreme weather events, for example, during flooding, extreme flooding, that can possibly overwhelm our water treatment works, for example, and maybe change and lead to these viruses ending up in our waterways and ending up in our taps. Very, very important. And then for extreme drought, for example, they can concentrate the viruses, so that with that concentration, again, it can easily be picked by hosts, and it ends up in our bodies. And then vectors, very, very important. With climate change, there is variation in areas where the vectors can colonize. Maybe the vectors were restricted to a particular area, but with climate change, these vectors can be able to colonize other areas. And that means we are bringing the viruses closer and closer to us. For example, an example of not enteric, but another virus that is vector-borne, the Zika virus. I know you've heard of Zika virus. Of course, it's more common in the South American region. So if maybe, it's mosquito-borne virus, that is. So if maybe another day you hear that all the mosquitoes from Brazil are going to be in Budapest, we'll all run away because we expect to suffer from Zika virus. It's a very bad virus, of course. You should read about it. So vectors and climate change is something that is real and it's something that you should worry about. Then another concern is the zoonotic spillover. I already mentioned this briefly, but I'll go back to it. Zoonotic spillover and ecological disruption. And again, this is because of various anthropogenic activities. We are clearing the habitat for various animals. We are interacting with these animals because we are replacing the green jungle with concrete jungle, of course, in very many parts of the world. And that means we are exposing ourselves to interactions with animals, leading to conflicts. And if transmission were to become systematic between animals and human beings, then of course there is going to be likelihood of spillover, like in the case of coronavirus, where people are talking about perhaps it came from a bat or from a food market, things like that. So that leads us to what is called One Health approach, which of course I have a representative diagram here. We're looking at the triad of human health, animal health, and environmental health. So it's a convergence of these three aspects where there can be cross-boundary contamination of the viruses. It can jump from human to environment to animal, and the circle goes like that. Zoonotic, spillover, and One Health approach. Then some other things is the environmental contamination and persistence. This is a very interesting area where, again, environmental viromics is being canvassed. And in this particular case, we are saying that, well, we have the waters, we have the waterways, but these waterways are every time being impacted by various pollutants, all manner of pollutants. Biological pollutants like pathogens, other pathogens like bacteria, for example. Emerging pollutants like microplastics. We have chemicals, we have nutrients. And basically, that contamination and viruses, there is something that we need to maybe explore here. For example, pathogen transport. Something like horizontal gene transfer. For example, biofilm formation as a result of pollution or pollutants, the biofilm formation has been found to be something that can retain viruses. And with that retention of viruses, there is higher chances of enhancing the horizontal gene transfer when we have bacteria and we have the phages, and of course, that is something that is realistic when it comes to biofilm as a source of pollutants, as a result of pollution. Something else is that some of these contaminants, for example, the chemical contaminants, they can maybe shield the virus from degradation. In other words, they can protect the virus. And when they protect the virus, they can make the virus be transported into various sections of the chain, and systematically we can end up with the virus where it is not supposed to be. Something very unique. Then it's also important to talk about the technologies here. And talking about the technologies, of course, since the era of electron microscopy around 1930s and 1940s, a lot has happened as far as technologies is concerned, which is making us to have a better way of understanding viruses in the environment. Classical case is the metagenomics, which has made us today, in terms of the history of viruses, We are in the age of viral dark matter. The viral dark matter in essence is simply meaning that we are able to unravel a huge set of viruses in the environment that are yet to be properly classified or are yet to be properly assigned function. So we have a whole lot of viruses that we don't know what they do, and we don't know their significance as far as being in the environment is concerned. The viral dark matter, they are yet to be annotated any function. Also, we have the digital PCR, which is something very unique as far as detecting viruses with very low concentration numbers in the environment is concerned. It tests with military precision, I should say. The digital PCR is very accurate in terms of detecting viruses and quantification. So in digital PCR, we can quantify viruses without necessarily looking at the standard curves, like in the case of qPCR, which I used a lot. In fact, when I was doing my research in the UK, we were approached by QIAGEN company to use digital PCR for what we are doing, because we are testing for viruses even in drinking water, the final drinking water, and because of the presumed low concentration, they were preferring digital PCR to qPCR because of that accuracy and precision. And so here is just a classical case of what metagenomics can do with this research in WHO, which was done in the People's Republic of China. And here they were doing surveillance in the groundwater in the People's Republic of China. So the color shades there is telling us of the sampling points. Of course, you can see in the whole of China, and the color codes there is mapping of the richness of viruses they found here, the ecological richness and diversity of the viruses they found in the whole of the country. Of course, with several annotated, which of course we see here when we look at these graphs. You can see there is an annotation even at higher scale levels of classification like realm and phylum. We have large number, the orange one is large number of annotated viral particles. Of course, the clear ones, I don't know whether you're able to see properly, those ones have been annotated. But even at the realm level, we should be able to tell all the viruses, should be able to annotate all the viruses at that level. Of course, we have here in the order family, we also have a few that are yet to be, we also have a lot that are unannotated and a few that are already annotated. So that's a classical case of what this metagenomics can do, of unraveling a whole lot of viruses without annotation. And here we have the train or the chain of classification of viruses, telling us that we are still lagging behind bacterial and even eukaryotes in terms of classification. And then finally, with regards to the conversation points, we have the viruses in the environmental processing, and this is something very important. It introduces something called the viral shunt, which is something that's relatively new. It's about 10 years old concept, the viral shunt, where we have a lot of activities that viruses do in the oceans and deep seas by breaking down the bacterial there and other biological entities like phytoplanktons. And when that happens, in that large breakdown, then we end up dissolved organic matter and particulate organic matter pool. And of course, what basically we're talking about here is that there is some level of retaining the carbon and energy within the sea, instead of letting this go up the scale of the food web, for example, to the phytoplanktons and the zooplankton and the fish. It is within the microbe world, such that breakdown make everything to be retained within the seas, and some carbon, of course, are sunk deep into the sea, maintaining deep ocean sequestration. So this is something that is about the microbial, we call it the microbial loop, where we have the recycling, the biochemical cycling maintained within the waters. Again, very interesting when it comes to viruses like bacteriophage, which works on the bacteria and other, and also we have phytoplanktons in the deep seas. So with that explanation, with all these emerging aspects, we set out to analyze viral ecology in Lake Victoria, and to do that, it's important first of all to understand where we are with our research. So we carried out this surveillance in Lake Victoria, which is one of the largest freshwater lakes in the world, supporting several people there. You can see to the tune of 40 million within the East African region. You can see where it is located within 3 countries. That's Kenya, Uganda, and Tanzania. A very large lake, of course, and very dynamic. So this lake is human intense, and human and water interaction is quite intense here. It's a source of drinking water for many. It is as an economic zone for many, like fishing, recreation. We have water transport and domestic use and all manner of things. We even have a park with several parks, not just one park, but several parks around the lake. So you can see how this ecosystem is very important, and because of that, there are multiple of contamination pathways that you could expect as far as viruses are concerned with this lake. So you can expect things like wastewater runoff, the surface runoff from maybe the towns that are surrounding there, the informal sanitation around the lake, of course, of the settlements, and even the diffuse sources of contamination from the catchment. All these things are real when it comes to this lake. So because of that high connectivity between humans, animals, and environment, this lake is like a natural lab for studying virus ecology and exposure interface, and that's why we dwell on it. Now, on to our aims for this particular phase of study. We are integrating virology, chemistry, and environment. In other words, we're going to look at virus-environment interaction in general here. And we had about 4 objectives. The first one was to characterize the virus occurrence at various points of the lake, with specifically targeting adenovirus and enterovirus. Again, we chose the 2 viruses based on the genomic patterns. These are 2 extremes when it comes to classification of viruses. We have double-stranded DNA virus, we have double-stranded DNA virus in adenovirus, and single-stranded RNA virus in enterovirus. 2 extremes. And so we wanted to compare the patterns between the 2 extremes as far as stability in the environment is concerned. So, in addition to that, we also set out to profile the chemical determinants in the lake, relative to the viruses there. So, for these chemical determinants, we targeted 12 chemical determinants. There are 6 heavy metals and 6 acid radicals. We'll get to see exactly which kind of chemicals are those. Then we also looked at the predictability. Here we are looking at the predictability of the virus occurrence and survival in respect to the concentration of these chemical determinants here. So we are looking at the influence of the chemical occurrence to the viruses. Then also we looked at the spatial and seasonal variability, because the area is very dynamic when it comes to seasonal changes with respect to rainy seasons and dry seasons. And so we had longitudinal kind of study to capture these kind of spatio-temporal variabilities as far as season is concerned. Now, to put it into perspective, this is the map, of course, map of Kenya. You can see there. You can see the lake is here, the darker one, and then the lighter one is Homa Bay County. This is one of the counties along the lake, which is one of the most populous counties along the lake, with a town adjacent to the lake. And so we're looking at the urban sources of pollution here. Then these are the sampling points. Hope it's a bit clear here. So we had 6 sampling points. So this is a settlement area. All this is a settlement area, and so you expect settlement, pollution sources of settlement here, and maybe the viruses from the settlement region. Then this site was adjacent to a sewage treatment works. So perhaps maybe sewage spillage as a source of pollution is what we considered here. Then there was an open ground generally where we had animals, domestic animals, grazing zone there. And then we have a fish factory, processing factory somewhere here. It's called Capital Fish Processing Factory. We're targeting to see if this could also be related to viral contamination. And then we had the CF here is for, that's the capital fish, the CF. The FM was a fish market. It's an open air fish market where, the women market sell their fishware from the lake to the local consumers. Then we have the landing zone. This is a pier. You can see a pier here. So it's a jetty where we have the ships from other countries. That's Kenya, Uganda and Tanzania, and boats from other regions within Kenya as well landing there. A hub for transport, for water transport. And then this other map is simply illustrating the lithology or the geology of the study area. Of course, there are a lot of mining activities, artisanal mining activities of various metals around the lake, and of course this is a lithology showing where these activities are taking place, including iron, including gold. And so in looking at the chemical determinants, this map is illustrating why it was a point of interest. Okay, the chemical determinants in respect to the mining activities. So of course we also have the pollution sources as maybe the carriers of these chemical determinants. So besides that, we also have the lithology or the geology of the area as a contributing factor. We had, of course, various processes here. I want just highlight briefly on this. Of course, the collection of water. That's me collecting the water. You can see there. Of course, I'm not going to fall into the lake. I know how to swim, by the way. So, in collection, we collected 216 samples across the 6 study sites, which means every study site was collecting 36 samples, water samples, in a campaign of 6 months, ending up with a total of 216 samples. Of course, the samples, because of the chances of getting virus in the water, they are very light in concentration. So in concentration, distribution of the water samples, so we have to do concentration process. And of course, in concentration process, we did 2 tire concentration because of that. The first tire concentration or primary concentration is using glasswood adsorption elution technique, followed by polyethylene glycol precipitation and sodium chloride precipitation at the secondary concentration there. Then, of course, we did a number of qPCR here. We had nested PCR. These are 2-tire PCR to increase specificity. Of course, the real-time, the reverse transcription qPCR and the normal qPCR, this one we did on the other side of the analysis piece in the UK, but this one we did back with the lake in Kenya. And then we also had water chemistry. And water chemistry here, we use a lot of spectrometry techniques to analyze the chemical determinants or the analytes for that matter in the water samples. Now, on to what we found out. Among the 216 water samples we collected, just only about 8.3% of them were contaminated with the 2 types of viruses. It sounds low, of course, but it gives us a snippet of what maybe a more elaborate scaled analysis could give us as far as contamination is concerned in the lake. Of course, of the 8.3%, adenovirus was 5.4, enterovirus was 3.4. As you can see there, adenovirus is dominating, and for a good reason. It's because it is more stable in comparison to enterovirus. Then, of course, as far as seasons are concerned, we did not detect anything that is so outrageous with seasons. They were more or less balanced. The difference was very, very small. And perhaps it is known that some of these enteric viruses are more inclined to specific seasons. But for this case, it was kind of plain. Perhaps because of the equatorial temperature, being that the lake is along the equator, maybe. So perhaps maybe that cancelled out everything, and so there was no difference as far as the wet season or the dry season is concerned. Then here, of course, these are the gels. These are just the gel images of our PCR products. We have Enteric Virus, we targeted the 5' and translated region at 143 base pairs, somewhere here. Of course, adenovirus, we targeted the exon gene at 138 base pairs, or at the second strain of primers, because this is a nested PCR. So the second strain of primers is where we have these figures. Of course, you can see we have the sampling, the samples, and of course the controls there as well. Then now on to the spatial dynamics of the detection. At the landing zone, of course, very low detection. The high detection was at the sewage treatment zone and at the residential area here. I hope you can follow the initials. I mentioned what they are the other side. Of course, here we have the residential area is here at the tip. There's the marking of red. Then on the sewage treatment area, it is here. You can see these are the stabilizing points of the sewage treatment. So you can expect that maybe there is spillage or remnants of the treatments entering the lake, and so the result is a bit high here. The other ones are low. And in terms of distribution of our target viruses, you can see adenovirus is 100% in the first three. Enterovirus is only 100% in the open ground where we had animals grazing. Then here, of course, you can see adenovirus is dominating even the other two. Now on to the chemical determinants. Of course, these are the exact determinants. We had 6 heavy metals and 6 acid radicals. And of course, this is a summary of the detection patterns in all the 6 sampling sites. So we had the means here, and we had the minimum detection for maybe a particular sample from one of the sites, and with the lowest here, and the highest, again picking one high sample from a specific point with the highest detection rate. And in this table, it only makes sense when we compare it with the World Health Organization limits for a classical or a normal open freshwater point, lake or river for that matter. So these are what is expected as far as WHO is concerned. And you can see that all of them were actually below what is expected, so that's good. It means that the water was fit for general use, not necessarily drinking, but for general use. However, you can see that iron and phosphate radicals were a bit elevated at certain point, just at the maximum. For example, iron at the maximum 0.8, again is 0.3, but of course minimum is very, very low. Again, phosphate here 0.9, again is 0.5. And perhaps iron is, because iron is the main metal around that region, which has been mined for years there, and of course the mining stopped, but there's still some artisanal piecemeal mining, so perhaps it explains why this high level is there. Again, phosphate is mainly because of pollution factors. For example, maybe chemical pollutants from the domestic system, or even from the enrichment with nutrients, even from the treatment work, water treatment. The sewage treatment works could explain this. And then on to the predictability here, we did modeling using binary logistic regression to analyze whether we can statistically correlate or statistically predict the future occurrence or the survival of these viruses in respect to the chemical determinants. And to test for that predictability, of course, we ran Hosmer and Lemmershall test, and of course it suggested that it was a good fit. As you can see, the p-value here is higher, so it means it's a good fit because it means that there was at any one point where the detection was not corresponding to, where the regression model was not corresponding to the observed patterns. And then for the Nagelcake, we also ran Nagelcake as squared, which we found to be 0.43. This is modest explanatory power, yes, and it's good because it tells us that 43 from 100 we have around, about 60 something, 67. something like that. And so it means that in the other side, there are some other confounding factors that could drive this kind of relationship other than just the chemicals. Something else that could drive the survival of the viruses other than just the chemicals in question. So it is somehow complicating something. And then generally, out of the 12, all the other contaminants did not show any relationship or did not correspond to the occurrence of these viruses, except these three. These three, of course, they showed weak but very significant association, beginning with cadmium here. Cadmium and adenovirus, you can see the odds ratio is about 1.28. So for every one unit change in the concentration of the cadmium, the potential of survival of adenovirus is 28%. Same to lead and enterovirus at 16%. And basically, we are, of course, the p-values you can see there. And basically, we are saying that this could be mechanistic, or it could simply be telling us that perhaps there is co-occurrence of, let's say, cadmium with the elements that are introducing adenovirus into the waters. Maybe there's some elements of co-occurrence. And then as for phosphate, there was a weak negative relationship with enterovirus. So this is a very interesting point. It's a negative one. As you can see, the odds ratio was very, very small here at 0.0001 there. And so it tells us that maybe there is some elements, maybe microbial activity introduced by the enrichment of phosphorus or the enrichment of the nutrients that somehow degrades the viruses, or there is something else. Maybe need to be investigated, but that's what we found out. That for every unit increase in phosphate content concentration, there is a decrease in potential survival of enterovirus. So basically, that's what we had in Lake Victoria, and so this journey will be incomplete because before we say what next, what do we have with this concentration when we talk about the engineered treatment systems. We will look at the ecological systems, but what of the engineered treatment systems? Because it's the same engineered treatment systems that abstract water from the ecological sources. The same water that we see in the lakes, rivers is the water we drink because it passes through the engineering system. So we continued with the journey of testing viruses now to the United Kingdom, and we targeted two rivers in the United Kingdom, River Trent and River Derwent, passing through central England. And so in this slide is just trying to elaborate why we are chasing this in the treatment works and seeing how viruses partition in the treatment system. Most of the such activities simply test the influent and the outfluent waters. But we went ahead to test in the specific compartments of the treatment system what happens. In an otherwise a highly sophisticated treatment system, one of the most sophisticated treatment systems in the world is that of the one where we did this. The treatment works is by the Severn Trent water, and the Severn Trent water is the one that in the whole of England is the main water treatment works, and they have treatment works everywhere, in every area. So in that treatment works, during our work, of course, this map, maybe I should explain this first. Here we have the map of the United Kingdom. The treatment works is somewhere called Derbyshire, is in the West Midlands, and this is the treatment point. So you can see it's beyond Birmingham there. Then since here is explaining the context on the rivers, River Trent and River Derwent, which are located around here. River Derwent you can see here, and River Trent should be also somewhere around there. Then now this treatment works. There were two treatment works that we were testing at the same time. In the same space, there was the conventional treatment works that has been there since the 70s, which was supplying the water to the domestic supply at that time. But besides that, they were also testing a more sophisticated treatment system, which was in operation, supposed to be commissioned this year. And this other test, the one that's being tested was more advanced one that was said to be able to even eliminate viruses from the final drinking water taps. So we had two treatment systems running at a time. The first one is the conventional one, which I've said has been there for some time, and is the one that's operational fully to the consumers. It's called Church-Wilne Water Treatment Works. And then we had, parallel to it, there was Witches Oak pilot plant, which is an advanced system introducing the concept of microceramic membrane filtration technique. It filters out even the finest particles in the water, and so it was thought that with the pilot scale of introducing the ceramic microfiltration system, it can remove the viruses. So that is the essence. We wanted to see whether this advanced one is also able to do something with the viruses, and it will be interesting to see what happened there. So in that case, our approach was stage result sampling. So every stage we were sampling, and then we quantified the viruses in the sample. So we wanted simply to understand how the viruses persist, partition, and attenuate across all these treatments. Now, for this particular case, we went beyond adenovirus and enterovirus. We scaled it down and introduced new enteric viruses. So we targeted norovirus G2. We also added rotavirus A and hepatitis A and E. So we had 6 viruses in total. So that is just a sample of the Church-Wilne treatment works, the conventional one that is operational, and these are the stages, and these are our sampling points. We sampled at the basin, at the clarification stage, of course at the source here, River Trent. River Trent is the only one supplying Church-Wilne, but when we come to the other one, it gets 2 supplies because of the volumes that are required there. So we sampled at those spots, gravitational filters, and then the gravitational filtrate of the effluent water now taken to the sewer, and then the sludge, which is as a result of this filtration. So the sludge, we also tested that, and then the granular activated carbon, which is the last stage before this infection, and then the final water that's now going to our taps. We also tested that. Then for the Witches Oak, all the advanced treatment systems, a more complex one, a good piece of engineering here, and so in this more complex treatment, they abstracted water from 2 sources, that is River Trent and River Derwent. The River Derwent was first, the water from River Derwent was first held in stabilizing ponds or lakes here as the first point of sedimentation before being mixed with River Trent, with River Derwent water. So here we have the river water treatment here. So we sampled the source, the combined Trent and Derwent, we sampled here. Then we sampled, this is where we have the membrane filtration, the ceramic membrane filtration technique here, which is built to be very powerful and able to even exclude the viruses besides the bacteria and the cryptos. So we sampled at pre-membrane and post-membrane. Then we also sampled the activation carbon, which was a new one being tested, and then they used activation carbon here. We also sampled that, and then after the activation carbon, which is the activation carbon is meant to remove the organic pollutants, for example. And so we sampled all that, and then we have the treated water. Now the treated water from here was not channeled to us because it was a pilot, so it was channeled back to the treated waste, to waste sewers. Of course, there's a pilot plant that is coming, subject to performance of this, which of course we will commission later. Now, on to what we found out. I'll move a bit fast. Here we had the Church-Wilne detections. So we had detections at every stage. So this is a description of the stages, and this is what we sampled. So detection here, adenovirus, here we used qPCR, and we have the CT values here, the CT values from the cycle values from the qPCR, cycle threshold values, I should say. So we have adenovirus at 28, 27 here, and norovirus is at 29 at the start. Now, interestingly, at the clarified water stage, we have adenovirus going up, and then there's an introduction of hepatitis A, but now we don't have norovirus. And then at the final treated water, there was adenovirus only, which is like a breakthrough. Then the effluent water, again, after the processing to the sewers, we had rotavirus being introduced. So, but generally what this is saying is that there is a nonlinear detection or nonlinear attenuation of the viruses from the start to the end, which means we could still have the viral particles in the waters. Of course, we did not detect, we did not test pathogenicity, but the viral genome is detected in the final waters, and that means either there is no absolute exclusion of the viruses in the treatment system, or maybe as a result of redistribution or the system holding the viral particles or flocculation patterns, we can still have viruses at different stages of the treatment works. That's what it says here. So you can see the pattern is from detection, non-detection, then downstream redetection. An interesting pattern which is not consistent with what you would expect. Of course, as for the sludge, there was no detection in the sludge, of course. That is what this table is simply telling us there. Then as for the yes, so here it's simply explaining why we think there was that kind of skewed detection patterns. Maybe, for example, we can say flocculation maybe promotes adsorption of the viruses to particulate matter, and that means it can be detected at a later stage. And these other factors could be explaining why there is no linear elimination or attenuation of the viruses, maybe hydraulic variability, operational dynamics, and the matrix inhibition, maybe in case of thick matrix or of the viral samples or on the water samples, that is. So, and then detecting viruses in the sewers simply means that we are introducing them back into the environment, in the flow channels in the environment. So this is just trying to explain to us. For example, we have detection, non-detection, then detection again in the final. Very inconsistent detection patterns. Then as far as genomic detections in the advanced strain is concerned, we do not have detections in the raw water. So it means the water we got from Debrecen and Debrecen, there was no detection. However, now in the pre-membrane feed, this is where I said there was introduction of advanced membrane treatment that works by self-exclusion principle that can even exclude viral particles. At the pre-membrane feed, we had adenovirus, but after the membrane, we supposedly have removed the viruses. The adenovirus, somehow it was partly attenuated. It reduced from 26 to 28. It somehow reduced, but as for the enterovirus, it was now introduced. So perhaps the holding of adenovirus in the postmembrane, at the postmembrane stage, was a result of the percolation into the treatment works, and then it is released into the waters at later stage. But overall, at the final treated water that was now to be taken back to the sewer lines, there was no detection. The last 3 stages, there was no detection. So maybe all the stages combined could possibly perform better. And what we are seeing here is that this treatment works somehow perform better as compared to the current treatment system. Even though very small difference, as you can see there. So here is simply explaining the potential reasons why, again, at the advanced level, the detections were not systematic. And then, of course, you've seen that adenovirus has been detected in both lake water and here in the treatment works. And this is simply telling us something about adenovirus as we've been following it. It was detected upstream and downstream, which means it was the only virus that breached all the treatment stages as compared to the other viruses. And so it persists, very persistent, meaning that when we started, we said that this is one of the viruses that's very resistant to treatments and even very stable in the environment. That perhaps explains why at Chatuine, for example, it started at 78, but at the final treatment, we still have it at 24. Though it was at clarified, here it went higher, but between clarified and the final, again, it reduced a little bit. of course. Then at Witches Oak, at pre-membrane it was 26, but it was attenuated a bit to 28, but still it persisted. So somehow it explains that adenovirus cannot be completely removed because of its stability and robustness. And so we need to understand some bit of biology about adenovirus, and we said it's non-enveloped, making it highly stable, and its double-stranded genome also makes it further to be stable in the environment, and therefore it is both resistant to physical and chemical stressors, more than the other types of viruses. So this is that structure of that virus. Of course, you can see we have the capsid here, the protein coats the capsid, and then we have the genome inside there, the double-stranded DNA inside here. It has got the spikes, which are used for clinging or hanging on the substrates, because this can be found even in fomites. So for hanging, attaching to the reservoirs for that matter, we have the spikes there. Adenovirus, very interesting virus here. So in conclusion, these findings are simply telling us that across the treatment works, you can see the viruses persist, especially adenovirus persists in both treatment works. So tells us that's resilient, and it can span both systems, both engineered system and ecological systems. Of course, other viruses are not as strong as adenovirus, and they were appearing sporadically. Now, as to what this means to water treatment and safety, the implications with water treatment and safety, it simply calls for improvement on the multi-barrier treatment works to ensure that at least we minimize as much as possible these viruses ending up into our taps. And then this need for virus-specific assessment, for example, targeting specifically adenovirus because of the special case, maybe specific attention to it. Then also need for monitoring strategies that can detect low copy detection. Of course, the inconsistency in detection perhaps could also be as a result of the low copy patterns of these viruses in the waters. So maybe there was the detection threshold that perhaps we did not reach in a particular sample, and so that could explain why there was a skip in certain sections. So this last point is simply telling us that we need to enhance our detection patterns, and perhaps this is when digital PCR can come in. So with that, I want to acknowledge the following. We had so many people that worked on this work. Of course, with my lead supervisor, Professor Ken Melitz of University of Nottingham. Also, I have Dr. John Maingi of Kenyatta University and David Velez of Southern Trent Water, who supported us with sampling and operational understanding of what we have in the treatment works. And Adam Lee helped in analytical work a lot. So thank you very much for listening. And I hope you love viruses, and perhaps I will have converts that can come and we study viruses together. Thank you very much. As for now, I'm extending this work now to look at the virus-host interaction in the environment. For example, looking at the environmental animals, particularly with respect to Hydra, to see the virus-host interaction at environmental level. Thank you very much.